Excellent. So now moving on to the next section, I'd like to reintroduce Jay to present T-cell class 1 tools, including binding, processing, and immunogenicity. Thanks again, Jay.
All right, remember me? Now I'm going to tell you about the T-cell class 1 tools, the binding, processing, and immunogenicity tools, and in particular, how you can take advantage of them on the new next-gen tool site. So we'll take a little bit of a deeper dive. And we'll start with an overview of the endogenous antigen processing pathway that probably many of you are familiar with. And what we're showing here is kind of a cartoon of a cell and trying to convey that these proteins are just constantly being degraded in this cell by the proteasome into shorter peptide fragments. Some of these fragments then get transported by the TAP protein into the ER and may undergo further processing and may bind to an MHC class 1 molecule, which then will make its way to the cell surface and may or may not interact with the TCR of a CD8 positive T cell. And we wanted to show you this because it helps to kind of delineate the different parts of the process that all of these tools are trying to predict. So for instance, the MHC binding tool more or less is predicting this part of the process. The processing tools are predicting the breakdown by the proteasome and transport by TAP protein. And the immunogenicity tools are predicting recognition by a TCR and an immune response. And so if we start with MHC binding, which again is this part of the pathway, we just come to the home page, or we can go to the next gen tools TC1 page. And like I showed you in the last movie, you can click on this link down here and you'll immediately get to the TC1 page with the example running, or you can click on this link and it will bring you to that page with kind of an empty slate. But before we go there, let's just talk a little bit about class 1 MHC molecule and its structure. So class 1 MHC is expressed by most nucleated cells, presents antigens to CD8 positive T cells, and it consists of two chains. So there's this short B2M or beta-2 microglobulin chain that's invariant, and then there's a longer alpha chain that is not invariant. It's highly polymorphic. And that's the chain that has the impact on binding. The binding groove is closed in this molecule, so it only accommodates relatively short peptides of lengths 8 to 11 amino acids. Bjorn showed a little bit about this yesterday, but just a reminder of where the MHC binding predictions come from. Like I mentioned, MHC molecules are highly polymorphic. They all have their own kind of binding preferences and the profile for the peptides that they like to bind. But that said, peptides may be able to bind multiple MHC molecules, and there have been a lot of experimentally characterized peptide MHC interactions coming out of Alex's lab in particular, and many other labs, and all of this data is in the IEDB, and all this data has been used to create these prediction models using machine learning algorithms. And so if we go to the T-cell class 1 page, and it's an empty slate, we have nothing entered in there, we'll just see that the example sequence kind of in gray, and we need to get our antigen sequence in there. There are a few ways to do that. You can type it in, you can copy and paste it, or you can drag and drop a file. And so I just showed an example of how we kind of drag and drop that same set of dengue envelope protein sequences in there, and we get a little bit of feedback down here that says, oh, I recognize this as a FASTA format, and there's about 2,000 characters. So as we continue on and show a little bit more of the page, the next thing to do is to specify the alleles and the peptide lengths. So the peptide lengths can be specified through the slider, and the alleles through the autocomplete or through the finder. And here I'm just showing two alleles just to make this page shorter and just for display purposes right now. But back to the reference set that I alluded to earlier, we have this reference set of 27 alleles, which covers about 97% of the population. You don't need to memorize this set. You can go to this link here and learn a little bit more about it. You can also, as I showed you, go to the allele finder and click on that 27 allele panel, hit submit, and have your prediction page populated with those 27 alleles. In terms of length selection, so the alleles differ in their preference for lengths in binding versus presentation. So on the left, we're showing kind of the in vitro binding length profile preferences for several alleles. And on the right side, what we're showing is the length profile of peptides that are actually eluted from the cell surface. And I guess the take home here is that these profiles look relatively similar on the right. These are the peptides that actually have a really good chance of being epitopes, and there's a big spike at 9 here. And so if you're going to predict for only one length, you'll definitely want to make that length 9. But you can predict for more lengths simultaneously on the LinuxGen tool site. And so if we continue with our example, we've entered our sequence, we've entered our length selection, our alleles, and now we're going to select NMHC-PAN 4.1 BA as our binding method. And just a quick note on the binding method selection. We currently have 2 recommended methods. One is the EL for epitope prediction, and the other is BA for binding prediction. Our recommendations have changed over the years. They'll continue to change as new benchmarks are evaluated and new data is obtained. But these are our current recommendations at this point. Speaking of benchmarks, and Bjorn talked a little bit about this already yesterday, we have established a weekly automated benchmark for binding, where new data that makes its way into the IEDB that's related to peptide binding MHC. We test each of these methods' ability to predict that data. And for the last year and a half or so, NMHC-PAN 4.1 BA has been coming out on top in this binding benchmark. You'll notice that NMHC-PAN 4.1 EL is near the bottom of this, but we're not predicting elution here. So this is purely a binding benchmark, which is why BA comes out on top. In other studies, we've seen NMHC-PAN 4.1 perform really well. So we've made all of our selections now, our sequence, lengths, alleles, and prediction model. The last thing to do is to click run. Once we do that, that's going to go ahead and take the sequence that was in the text box, break that up into all possible peptides of the chosen lengths, unless that as-is option is selected. So the as-is option gives you the ability to, instead of pasting a antigen sequence in the text box, put a list of peptides in there, and those peptides will not be broken down any further. They'll be predicted at the length that they're entered in that box. So once we have our peptide list, each of those peptides binding affinity and elution score or elution score is predicted based on whichever method is selected. Those predicted affinities and scores are then compared to those of a set of randomly selected peptides to calculate a percentile rank. And if you do select multiple methods, you'll also get a median percentile rank back. So like I showed in the movie, once you click play or run, you'll get a screen that looks something like this with this loading menu, or sorry, this loading graphic. But eventually you'll get a table that looks something like this. I've gone ahead and removed some of the kind of superfluous columns here just to show you the columns that are more related to binding, so the NMHC-PAN VA method that we selected. So again, we'll have one row per peptide and allele, and then columns related to all of the different tools that we selected. So here we have the NetMHC-PAN VA IC50 and its associated percentile rank. And just to talk a little bit more about percentile ranks and IC50 values, the IC50 value distributions can vary pretty widely from allele to allele. And so what I'm showing here are the predicted IC50 values for 10,000 randomly selected peptides that we use to rank, to calculate our percentile ranks. And this is for HLA-A0201. On this side is where you would see the binders, and you can see most of the peptides are not binders, right? They're on this side of the distribution. This is a typical curve that we would see. But if we look at HLA-A2402, the shape of the curve looks the same, but we can see that it's shifted kind of way to the right of that of HLA-A0201. And so it can be hard to compare directly the IC50 values between the two alleles. And here's our typical 1 percentile rank cutoff. If we kind of zoom in on that section of the plot, it looks something like this. And so for HLA-A0201, the first percentiles at about an IC50 value of 27. For HLA-A2402, it's around 500 or so. And so by mapping those IC50 values onto a percentile rank, it makes it easier to kind of compare the relative binding affinities of the peptides among the different alleles. If we go back to the output, we, like I showed you in the movie from the last presentation, there's this display columns button that will allow you to get a description of all of the columns and select or deselect columns from the display. As you select more methods, you're going to get more columns. So here, if we additionally add an NMHC-PAN EL and SMMPMBEC, we're going to get more columns in the output. And this functionality becomes more important because you probably want to remove some columns that are just less important for your purposes. And also get a description of columns that you might not understand the meaning of from the titles. So you can also download the results in different formats, TSV, CSV, and JSON. If you download a TSV or CSV, it'll look something like this, and you can open that up in your favorite spreadsheet program. Like I showed you earlier, also you can enter your email address and a name for your job. And when it's completed, you should get an email that looks something like this, that will have the name of the job in the title, and it'll give you a link back to the pipeline where you can view the results. And you'll also get the result as JSON at this point. I think we can update that to TSV or CSV as well. So we've done our prediction, and we have our results. And now we want to know, how do we select the peptides that we want to move forward with? And there's no right answer. There are many different methods that we've used in our groups over the years, including just picking the peptides that are below percentile rank 1.0. Similarly, picking the top 1% of peptides for each allele and length combination. Both of those methods will give you relatively equal numbers of peptides per allele. You can also use a hard affinity cutoff, for instance, 500 nanomolar, which corresponds to intermediate affinity. That will ensure that all the peptides have a reasonable affinity. But because of the different IC50 value distributions, as I showed you, you will end up with a very different number of peptides per allele, if that's a concern. There's also allele-specific binding affinity thresholds that have been established by Bjorn and Alex's group a few years back. And you can find out more about that through the tools documentation, where we talk about the thresholds and interpreting scores. And we link out to there's a Zendesk article that we link out to that has even more information on the allele-specific thresholds for class 1. And then here's a link to the journal article where these were determined. And there are 2 recommended thresholds, one for capturing 75% of the binders and another for 90%. And so, I mean, to summarize the recommendations, there's no one approach. All of the approaches are reasonable. We've used many different approaches in our studies. You can combine thresholds, for instance, take the peptides in the top 1% and with less than 500 nanomolar. And there are some allele-specific thresholds that you might want to use as well. All right, so that's binding. Let's move on to processing, which again is this part of the pathway. And we go to the same website, same links, the T-cell class 1 page, everything is on that page. And we come here, and now instead of selecting a binding method, we're going to select processing. On the legacy site, this is called our combined predictor. On the next gen site, it's called our basic processing predictor. And what it does is it will ask you to choose a MHC binding method because it needs an IC50 value to run this prediction. And it'll calculate the MHC binding using that method. And then it'll also give you a likelihood of proteasomal cleavage for that peptide, as well as tap transport. And if we zoom in on that section here, so these are kind of the method-specific options. You'll get options for proteasome cleavage. So what kind of motifs are we looking for? Are we looking for motifs that are used by the immunoproteasome or the constitutive proteasome? And there are also options related to tap transport. But our recommendation is really just to leave those at the defaults, especially the tap transport options, unless you have a very specific reason to change them and you have a very good understanding of the algorithm. Just leave them unchanged. They're just provided there for convenience. So, sorry, my Zoom controls are in my way. Right, so we have, again, selected our, entered our sequence, done our length selection, allele selection, selected the processing method, and now we click run. And then we'll get a table that looks like this. Again, I've removed kind of superfluous columns here and just want to focus on the columns that come out of the processing tool. So we get columns for proteasome score and tap score, higher is better. MHC score, which is now a negative log transformed IC50 value. So again, higher is better. And the processing score is now additive. It's just the proteasome plus tap. And then this total score is going to add on to that, the MHC score. And so in terms of recommendations for how to use this tool, processing is much less of a limiting step than MHC binding. Most high affinity binders are also processed due to the coevolution of these molecules. And so we recommend to use this tool more as an investigative tool to determine maybe you have a peptide that seems like it's a good binder, but it doesn't seem to actually be a good ligand. Maybe it doesn't have good processing score, or also potentially use the total processing score just as an additional filter for with which to rank your peptides. And as an alternative, we recommend to use methods that are trained directly on eluded ligand data, such as NMHC-PEN-EL, that have the advantage of the peptides that they're trained on are peptides that have gone through most of that endogenous antigen processing pathway. They've been processed, they've bound to MHC, they're made their way to the cell surface. The only thing they haven't done is been recognized by a TCR. But these have a very high, much higher likelihood of being in epitope because of that. And we just wanted to throw this in there. This is not on the next-gen tool site at this point, but we have had developed another tool and released it last year called AXEL-F. And what AXEL-F does is it takes not just the binding affinity of a peptide into account, but also the expression level of its antigen, of the source protein that it comes from, to give a likelihood score for whether or not this can be eluded from the cell surface. And it's based on the observation that peptides that have even poor or modest binding affinities can kind of overcome that if they're very highly expressed and be observed as being eluded on the cell surface. So I invite you to take a look at that tool as well. The last tool in the T-cell class 1 suite that we wanted to talk about today is the immunogenicity prediction tool, which again is the last part of this pathway. And again, same page and the same links. And the immunogenicity tool is based on a data set of based on two data sets, one of immunogenic peptides and one non-immunogenic, and studying the amino acid frequencies within those two data sets. And what we observed was that within the immunogenic set, we have an enrichment of amino acids like tryptophan and phenylalanine and isoleucine. And in the non-immunogenic data set, we have an enrichment of serines and methionines and lysines. And then we can use that data, we can use those enrichments to calculate propensity scores for peptides as to whether or not they're likely to be immunogenic. And so this is what a prediction, what the inputs would look like, very similar to the other tools, except now you're selecting immunogenicity instead of binding or processing. And you get a selector that lets you select which positions you would like to mask. So which positions do you not want to include in the calculation? And generally, these are going to be anchor positions. There are options there so the default is position 1, 2, and the C terminus. There's also an option to use an allele-specific sequence mask. And you can enter a custom mask as well. We're just going to go with the defaults here and hit run. And this should start to look really familiar at this point. This is what the output's going to look like. The immunogenicity tool only has one output column. It's this immunogenicity score. And higher is better. So higher scores are more likely to be immunogenic, lower scores less likely. We generally recommend a cutoff of 0 for this tool. And just one word about the performance of this tool. So on the right-hand side here, we're showing a set of epitopes and non-epitopes and their corresponding immunogenicity scores based on this tool. And we can see the distributions are different, but they're definitely overlapping quite a bit. But this tool can discriminate between epitopes and non-epitopes with an AUC of about 0.65 to 0.69, depending on the data set. So that's good, but it's not nearly as impressive as the AUCs that we achieve with our binding and the EL tools, which are in the 0.95, 0.97 range. So again, our recommendation with this tool is to kind of use it as another guideline with which to rank your peptides and taking the performance into account. All right, so we did all of our predictions individually. We did an individual binding prediction, we did a processing prediction, immunogenicity prediction, but we didn't have to do that. We can do everything all at the same time. And so that's what we're going to do here. Again, we're taking our dengue envelope proteins from 4 different serotypes, and we have selected our peptide lengths from 8 to 10. We're using our full 27 allele panel, and we're starting off with NMHC PAN 4.1 EL. And we just have to click on this little drop down to add another prediction. So let's go ahead and add our processing method with NMHC PAN BA to calculate the IC50 and our immunogenicity prediction. So we have a representative of all 3 methods there. And we click run. And that what we just did there was a relatively large prediction. So 27 alleles, 3 lengths, and 3 or 4 methods. So it was over 230 predictions that get sent to our cluster and broken down into that many jobs. And this one takes about 2 to 3 minutes to run on the cluster. And when it's done, you have this table of about 157,000 rows. And then you'll probably want to do some filtering. So this is the same filtering that I was showing in the previous presentation. We're going to filter by median binding percentile or 2%. And then we actually, I think I also added a filter here for immunogenicity score greater than 0, processing score greater than 0. So now we have trimmed that down from 157,000 to our 276 candidates. And just as a coincidence, yesterday this popped up in my news feed, and I thought it was just fortunate that the number 276. So cats have 276 different facial expressions. There must be some connection there. So then you have your data that you might want to move forward with, and Bjorn is going to show you how you can chain that into pipelines, send that to another tool. And you can also, as I showed you earlier, go to the pipeline map and pull out the link to this pipeline. This top link is going to include all of the data, the input data, the output data, including all of the filters that you set. And this second link is only going to include that pipeline configuration. It's just going to include kind of the algorithm portion. So just a quick recap of what I showed you. Our next-gen tool site it has that complete T-cell class 1 suite, which consists of MHC binding and elution, antigen processing, and immunogenicity tools. Our recommended methods have changed and will continue to change as the new algorithms are evaluated. We don't have a uniform threshold for selecting peptide candidates. There are many reasonable approaches, and for many that we haven't thought of ourselves. And the tools that we have, you don't have to execute them one at a time. You can execute them simultaneously, and you can filter your results, and you can share your results through those pipeline links. Thank you.